and we'll move swiftly on to Dr. Mel Lacey from Sheffield Hallam.
Hi, thank you very much. Okay, so I have said that I'll talk today about some microbiology labs that we've been doing on Zoom, which we really enjoyed, and we've had really nice feedback from the students of, so I thought I'd share it with you guys here today. So I'm a microbiology lecturer and I work at Sheffield Hallam University. So the background to this is sort of what we did before, I guess. So we used to do this practical class that sort of ran in two parts. The first part is within the lecture theatres, we used to get our students to inoculate plates, and we used to take them away, incubate them, and then they'd come back to the lab for a lab session to analyse their plates, but also, depending on what course they're on, perhaps do some further analysis on those plates as well. And the courses that did this sort of intro microbiology lab stroke lecture practical were our biosciences students, the nurses, and the ODPs. So the ODPs are operation departmental practitioners. So I need to say my thank yous here because I always forget to say them at the end of a talk, so I get really excited about the questions. So in the past, I've co-taught these labs for the nurses and the bioscience students because they're really big cohorts of between 250 and 400 students in a lecture theatre. So me and the lovely Tim Nicol have always done them together, and we've carried on doing these together online as well. So I just wanted to thank him at this point and let everybody know that he's helped me design them all. So what we did in the past was we gave students 3 agar plates in the lecture theatres, and we let them swab an object that they thought would have a few bacteria on, and then a lot of bacteria onto one plate, do skin swabs for skin low and high, and then do them hand washing, so before and after, and we've got different parts of the lecture theatre to wash them, their hands with soap or with hand gel, and then they'd go to the labs and then analyse them. So obviously along came COVID and we've had to alter how we teach, so we decided that we really enjoyed this practical and the students got a lot out of it, as we've adapted it to Zoom. So the first thing that we've done is we've got the technical team to provide us with some plates and some swabs. We've been able to carry on running some of our labs on campus. So both Tim and I have been going in and out of work perhaps once a week. We've been able to collect things from the technical team, bring them home, store them in our fridge, and then do stuff with them and then take them back to work to be incubated. We've co-taught this over 2 1-hour Zoom sessions, and we've done hand washing plates. We've added in coughing onto plates with and without masks, because I thought it was topical. And then the part that the students enjoyed the most is we let them vote where Tim and I swabbed using Zoom quizzes. So these are the examples then. So this was in the first session of sort of the votes that we let them do. So we sort of did each one individually. So we had the first vote, which was where they thought an object that they thought there wasn't going to be very many bacteria on. Then we did that plate, and then we did a high number of bacteria, a low number of bacteria on the body, and these were things that they'd chosen to do when we'd done the lectures, so we knew these are the kind of things that the students were interested in. And then the final one we did was an area of the body that had a high number of bacteria, and we let them vote. So full disclosure, this did lead to me showing my belly button to 250 students in close-up whilst I was swabbing it for the plate, and I still go a little bit pink thinking about it, but they genuinely thought it was absolutely hilarious. So, you know, it's hard for them all sat at home, isn't it? So I'm glad that I can provide that light entertainment for them. Yeah, so then we took them into work. I've gone pink just thinking about it, actually. And then we went into work and we incubated them up. And then because we've been in and out, we've been able to get back into the labs and photograph them all. And then in the second Zoom session, both Tim and I, again, we co-taught a session that was between 1 to 3 weeks after the first session, and we shared the results. So this is an example of one of the sets of plates that we showed the students. This is my cough plate without a mask and my cough plate with a mask. And as you can see, like I'm quite proud of these pictures. So there's quite a lot of microbiology in here. So these are blood agar plates. So you can see some of them have got like zones of hemolysin around them. You've got different colony morphologies, different colony sizes. So there's like a lot that we got out of these in the sessions. So Just to give you like the full experience of what it's like to be in my life, I'd like you to vote on, so I've got some, what's the word? I've got some pictures ready. Now, I've only shown you my AGAR pictures. I didn't really feel it was fair for me to show Tim's without him being able to be here to, you know, represent himself. So if you could do me some emojis. So if you'd like a leg, can you give me a little clap, a computer screen, a whoop, a thumbs up, a phone, a heart, inside my mouth, a little cry, and then a belly button up, and I'll have a look see. If you can't do the emojis, can you pop it in the chat? I'll have a quick look. Okay, I've got a belly button. Of course, I've got a belly button. Any more? Oh, okay. I think phone might win, guys. I was really expecting to have to show you my belly button. Okay, great. Okay, so this then is my phone plate, which is a little delight. I always like to, what's the word, make an argument when I show people my reading rank phone that I've got small children, and that makes it better, right? Yeah. So this is my phone plate that they just thought was hilarious, and you just see some really nice, yeah, hemolytic colonies there. All right, so what then was the outcomes of these sessions? So in the 2 1-hour sessions, we covered all the material that we would have covered in the lecture as well as in the second microbiology lab. So in terms of all the microbiology background, and then the practical stuff, like I said, you know, like the colony morphology and the differences between colonies and lawns and all that kind of stuff. We had really, really good lecture attendance online, which was really lovely. And we had extensive use of the chat, like they were on it like straight away, just making comments about the plates and the swabbing. And that led to a lot of really relevant questions and really relevant discussions. And a lot of that was really course specific. The nurses, for example, asked a lot of questions about COVID and sort of like how could you catch it and can it get in through your skin, whereas the biosciences were much more interested in sort of the mechanics of how the plates worked and things like this. And it was a lot of fun. I was going back through the recordings that we made of the live sessions for the students that couldn't be there, and there was just a lot of like laughing and jokes. It was just really nice, like light-hearted session. I haven't got any feedback from exams or anything like that at this point, and we haven't done our formal mid-module reviews yet, but the feedback we got from the students we've had so far was really nice. So they said it was an amazing session, they engaged throughout, they couldn't wait for the next session to see their plates, and my favourite one was, who knew that I could be so interested in microbes? Who knew they could be interested in microbiology? That's nice. Yeah, so I was like, that's a convert, so I was pretty happy with that. Yeah, so then just our conclusions for doing this was, yeah, it was fun and it was informal, and it was a little bit scary, like swabbing all bits of you and showing students, but they thought that they all took it in really good humour, and that was really nice. We had really good engagement, really good student discussions, and the reason that I wanted to share it with you guys today was that I thought that it would be quite easy to transfer to other labs and other sort of practicals where you're sort of doing sampling. I was thinking like dipsticks and urine and things like that. Obviously, we risk assessed all of this, and the people that I live with kindly allowed me some fridge space to store the plates. So obviously that is a requirement as well. So yeah, there you go. Thank you very much. I'm going to do my little questions one. Thanks.
Yeah, thanks, Mel. We're passing that on to our microbiology teaching teams. I'm sure they'll be keen to adapt that. I've had a couple of questions that come through in the chat from Lewis. How do you identify 400 plate photos and get them back to the right students?
In real life, when we did it in pre-COVID times, what we used to tell them what lab class they were in before they came in, and put the pieces of paper on the floor in the lecture theatre. So they had to put the pieces, the agar plates on the pieces of paper, and we used to wrap them up and put them in boxes to incubate and then bring them out at the right times. But obviously now we just did it with mine and Tim's plates, so we only had a little box full. So yeah, it was really, really straightforward. Yeah.
Ian said, I really like this, and the direct link between remote and online campus experiments sounds like a lot of fun as well. which I think we can all echo. And David said, can you see this being used for other show-at-home experiments on other topics?
Yeah, so I was thinking about this, and I think that you could do it quite easily with, and obviously, like, I'm a microbiologist, so I obviously go to micro, but things like environmental biosciences and looking at antibiotic resistance in soils, bacteria, like, you don't have to get plates out for all the students. That's like, obviously, yeah, but just getting plates, like, so me and Tim were the only people that had plates, and they were voting on what we did. So just having you and another person, and you can be like, okay, well, this is my house, this is someone else's house, these are the soil samples we've got, these are the, you know, antibiotic resistance we've found in them. You could do, like, if you're feeling brave, you could do, this is my urine when I woke up this morning, this is my urine when I went to bed, or, you know, like, and do dipsticks to see, like, different levels of things come up. And so, yeah, I think that they liked it as well because it was quite personable. You know, me and Tim had a lot of banter, because irrespective of what area of the body we do or what part of anything we swab, my swabs always come out with so much more crawling than Tim's, and they just think it's hilarious that I'm always just like, and here is my plate that is more horrible than Tim's, and it's because he didn't swab it so well, or I just have an outie belly button and that makes it different, you know what I mean? So I think that they liked the fact that we were being human beings rather than just, like, giving them loads of information all the time. So yeah,
I always love it when we're self-depreciating as well.
yeah.
David said, I'll have to show you the differential centrifugation video I did in the garden. You can share that at the end, David.
Yeah, I'll dig it out. It's quite something. I nearly killed myself.
Yeah, you're definitely sharing that at the end. And Ian said, I guess the approach could be adapted in the lab as well. Just the experimenter in the lab being filmed and students on the screen directing the action may help the limit on lab numbers. I think that's a really good idea, Ian. I think it's interactive labs is definitely something that a lot of people are striving for. So that's a really nice way of doing it. Fantastic.
I also at one of these lab sessions, I had my 8-year-old boy at home self-isolating, and he was doing the swabs, and the nurses were like, get your son to do this, get your son to do this. And, like, yeah, it was like everybody had a great time, including the 8-year-old. So yeah, they enjoyed bossing him about, and he enjoyed complying completely. So yeah, thank you.